Hello, everybody. Back to the Public Health Applications course. We started off the morning with some insect population ecology from Sergio Estay, and I think that's a very nice complement to what we've been talking about because it talked about the mechanics of population ecology in the face of changing climates. Now we're going to shift into a series of 4 worked examples, essentially case studies of the sorts of things we've been talking about. We're going to start with Lindsay Campbell speaking about Buruli ulcer in West Africa. Lindsay is a doctoral student here at the University of Kansas. She has a master's in quantitative geography and is working on several different disease systems for her doctoral studies. So I'm going to turn you over to Lindsay.
Okay. Hello. As Town mentioned, I'm going to be talking about Buruli ulcer disease today. So I'll go ahead and pull up my presentation. Okay. So Buruli ulcer disease is a, looks like we're having a little bit of trouble. Okay. Buruli ulcer disease is a necrotic skin ulcer disease that's present predominantly in West Africa. It's an environmental pathogen caused by Mycobacterium ulcerans, which is a mycobacterium that's in the same family as leprosy and tuberculosis. And it's a mycolactone-producing mycobacterium, which means that it has a lipid toxin that is responsible for attacking fatty cells. It's classified as slow-growing, and it has an immunosuppressant property, which goes undetected by the body. So this enables ulcers to spread over large areas of the body without any pain. There's 3 different stages of the disease. It starts off usually with a painless nodule or plaque, and then it follows by a small ulcer. And then if it's left untreated, then it can turn into a large ulcer. Treatments include, in early stages, antibiotics, which have been effective as long as an ulcer is not present. But usually patients need to have surgery, and the surgery is often invasive, and a lot of healthy tissue has to be removed around the ulcerated area. In severe cases, there's amputation of limbs. There's also several complications that are associated with Buruli ulcer disease, including osteomyelitis, which is when the infection gets into the bone, secondary infections. People often lose mobility because of complications, surgery, or scarring, and there's a high recurrence rate for the disease. So BU is present in at least 32 countries worldwide. As I said before, it's predominantly found in West Africa in children under the age of 15. It's classified as a neglected tropical disease and also an emerging infectious disease by the World Health Organization. It's hard to know exactly how many cases there are of Buruli ulcer disease because there's chronic underreporting across the entire region, and this is often due to misdiagnosis, lack of access to healthcare facilities, and there's also a strong negative social stigma associated with the disease. So lots of people are reluctant to seek medical care. So other challenges with Buruli ulcer disease is that the mode of transmission is unknown. So one hypothesis is that people get abrasions in their skin and the bacteria enters through these abrasions. And some people are researching the possibility of insect vectors that may be either mechanical or biological transmission. But if this is the case that there's an insect vector, it would be the first mycobacterium known to be transmitted by a vector. Additionally, the ecological niche of the bacteria is unknown. There's been several anecdotal cases linking human cases to flooding events or land cover disturbance, such as gold mining activities. but most of these studies have occurred at a very local scale. And so we wanted to go ahead and take a look, kind of a step back, and try to look at environmental factors that might be related to Buruli ulcer disease across the entire West Africa region. So the objective of this analysis was to predict potentially suitable environments across West Africa using a correlative modeling approach. So we started with this occurrence data set, which was acquired and cleaned and georeferenced by our Belgian colleagues. These are all laboratory confirmed cases from 1997 to 2006, and there were a total of 831 unique communities that we were able to georeference. This is actually a pretty exciting data set to get to work with because it's probably the only data set that crosses several countries, and it's the most contiguous and the most comprehensive data set available right now. But as you can see, there's a lot of cases reported in Benin, and this is not necessarily due to Benin having a higher number of cases than, say, Ghana or Cote d'Ivoire, but it likely has to do with the fact that they have a really robust active surveillance program, and so nearly every case of BU is reported in Benin. But for our modeling purposes, this introduced a sampling bias. And so we decided to go ahead and divide our data set into 3 different data sets of random points with approximately 275 locations in each data set. And then we would run our models on those data sets individually. For environmental data, we decided to use the MODIS NDVI and land surface temperature data at a 250 meter resolution. The NDVI is basically a measurement of greenness in the environment in vegetation, but it can also be used as a proxy for soil moisture. And so that was our interest in this product for this analysis. These data were acquired from July 2002 to December 2006. And this was because the July 2002 was the earliest that the data was available. We downloaded all of the data through the NASA Reverb Echo data portal, and all of this data is available for free. We've put together a tutorial on how to download the NDVI product and to process it using the MODIS reprojection tool. And so we'll post this to the website. And so anyone who might be interested in exploring this data further will have kind of the basic starting points to process the data. There's also a lot of additional data products available through this website. And depending on the research question that you're asking or your region and time period, you can really put together a nice comprehensive data set if you start to explore some of these other data products. For our purposes, because there's so little known about the ecology of Buruli ulcer disease, and there's several lag times associated with the disease in individuals, we decided to go ahead and just make a composite of these 2 data products across our entire study period. And in order to do this, we use local cell statistics. And anyone who's not familiar with this process, I've put an example of how to calculate the minimum local cell statistics in the graphic on the right. So if you look at the bottom left-hand corner of each of the raster layers to the left of the equal sign, you'll see a 4, a 3, and a 1. And 1 is the minimum value out of those 3 data sets, and that's the number that's placed into the corresponding cell to the right of the equal sign. So this is the process that we followed for each of our data sets, and we calculated not only the minimum value for each cell, but the median, maximum, and then the range between the minimum and maximum cells. And this resulted in a total of 8 raster data sets, and these were the environmental layers that we put into our model. We also had to come up with an M hypothesis for our calibration region. And in order to do this, it was a little bit challenging because often you kind of think of mobility when you're thinking about generating a M hypothesis, but with an environmental bacteria, and especially when you don't know much about its ecological niche, we had to go ahead and go about this a little bit differently. And basically, we decided to take a look at the World Wildlife Fund ecoregions of the world polygons that represent different ecosystems and environments. And we looked at the polygons that covered where our occurrence data were located. But we also wanted to provide a little bit of an environmental buffer as kind of a transition zone to make sure that all of our occurrence data fell within these polygons. And so the green polygon on the right represents our final calibration region. And then the black dots are our occurrence data from our first subsample data set of randomly selected points. Here's another view of our calibration and projection regions. On the left is the median in DVI. The area that is green represents more densely or denser vegetation and likely higher soil moisture. Yellow areas represent drier regions. And then within the yellow square across the landscape is our projection region. On the right is the land surface temperature data. Red areas are higher median land surface temperatures, and blue are cooler land surface temperatures. So after generating our environmental data sets, we were ready to go ahead and run some models. We decided to use Maxent. And as I mentioned before, we had 3 individual randomly selected occurrence data sets, and we ran models on each of these. We used a jackknife to take a look at all of our variables to reduce some or remove some variables that might be redundant, but also to see which variables were contributing to the model performance the most. And in this case, the land surface temperature data variables seem to be the major drivers of the model, especially land surface or maximum land surface temperature. We also thresholded our model with an error of 10%, and we validated our model using our withheld data sets. And in order to validate the model, we performed a cumulative binomial test, which predicted accuracy significantly better than expected by random, and we had a p-value of less than 0.01. We also wanted to take a look at the potential for model extrapolation, so we performed a mobility-oriented parity test, or MOP test. So this is a picture of or a map of our projection and results. If you look at the red areas, those are areas that have been predicted highly suitable for Buruli ulcer. And then yellow regions are less suitable and gray are unsuitable. Within individual countries, Sierra Leone, Cote d'Ivoire, and Togo had the highest overall percentage of their land mass predicted as potentially suitable habitat for BU. And then in terms of just the actual number of hectares, Nigeria, Cote d'Ivoire, and Cameroon had the highest number of overall hectares. As I mentioned a minute ago, we performed a mock test on the data, and the thing to pay attention to here is locations where you see black squares. And basically what this means is that the combination of environmental values that are located within these black squares were not present in our calibration region. And because it's a correlative model, when we're predicting to that area or that region that has those environmental combinations, we can't be as confident about the prediction at that actual location because the model is likely extrapolating. So if you'd like to read more about this test and how you might be able to use it with your data set, our ecological niche modeling group wrote a paper to the left, The Constraints on Interpretation of Ecological Niche Models by Limited Environmental Ranges on Calibration Areas. And this is basically an extension of a test that's available already in Maxent. So back to our prediction map, you can see once again, there's quite a bit of area that's predicted potentially suitable for Buruli ulcer disease. Obviously, within these areas, certain things have to take place, unknown behavioral activities, or a combination of events for a person to actually become infected with BU. But because so little is known about this pathogen, this, I think, provides a very nice tool to kind of start with. It could be used for people to narrow in to more specific regions, maybe to assist in potential field site for research, or for public health centers to investigate further for active surveillance. And it also helps across regions that do not have active surveillance. For example, Nigeria. It'd be pretty interesting to hear what our colleagues there have to say about our prediction. So in conclusion, this is, like I said before, a pretty exciting data set to get to work with. It's the first time that we've been able to look across several countries at once and kind of get a regional look at BU. In the future, we'd like to incorporate more data. We know that Buruli ulcer is taking place in the Democratic Republic of the Congo. I think if we add this data to our overall data set, then we would be able to calibrate our M region a little more accurately, and that will help to reduce those areas of extrapolation by the model. And we also would like to explore some additional variables such as soil, different soil properties or precipitation. And we want to continue working with everyone across the world and in different disciplines. So with that, I just want to acknowledge our colleagues in Belgium, Patrick, and then Andres, Town, and Yoshi here at KU and CDC and the KU Ecological Niche Modeling Group.
Okay, thank you very much, Lindsay. That was our first case study of the day, and we will be back online in about 5 minutes with a second case study.